Hi everybody and welcome to lesson 2 in section 1.6, which is looking at chromosome mutations. And in this lesson we're going to describe what chromosomal mutations are and the impact they have on the organism. By the end of the lesson I want you to be able to identify what the 4 different chromosomal mutations are and describe the impact they have on an organism. So our chromosomal mutations are a little bit more severe than our gene mutations. So rather than influencing just one nucleotide and changing one nucleotide within the sequence, within a chromosomal mutation, it involves the breakage of one or more chromosomes. So we're impacting on more than one gene often. So it's not just a single nucleotide within one gene. We're often influencing, several genes at the same time. Now, just like I gave you a little word to try and remember the different gene mutations, we're going to use the word tiddler here to remember our chromosomal mutations. So if you remember Tiddler the fish, if you read those books when you were younger. So the reason why I say to remember the word tiddler is because the 4 chromosomal mutations that we're going to take a look at are translocation, inversion, deletion and duplication. So inversion, not to be confused with insertion. This is translocation, inversion, deletion and duplication. Now, before we take a look at the different chromosome mutations, I want to take a little bit of time to explain to you about what a homologous pair of chromosomes is, because you've not really taken a look at this so far. So within the cell, if we're looking in a eukaryotic cell, in the nucleus of a eukaryotic cell, remember that we have linear chromosomes. Now, in a human eukaryotic cell, our DNA is arranged into 46 linear chromosomes, and they are arranged in pairs. So you can see here on the right hand side we have a picture of the human chromosomes arranged in their 23 pairs. The reason why they're in pairs is because you inherit one set of chromosomes, so one set of 23 chromosomes from your mum in the egg cell, and then you inherit one set of chromosomes, so one set of 23 chromosomes from your dad in the sperm cell. Okay. When they join together to form the zygote, those chromosomes are arranged in homologous pairs. So on the left hand side you can see a picture of a homologous pair of chromosomes. Now this is not to be confused with the sister chromatids. So remember, we often see our chromosomes as little X's because chromosomes, remember, only appear just before a cell divides. Remember, we want to package all of that DNA into these little suitcases so we don't lose any information. However, the difficulty is prior to cell division our DNA has replicated because we need 2 sets of DNA that we're going to split into our 2 separate cells at the end of division. So you can see here the chromosome consists of 2 sister chromatids. That's because each chromosome has 2 sets of the DNA. However, a homologous pair of chromosomes is basically a pair of chromosomes because one of them has come from mum and one of them has come from dad. Those chromosomes will contain exactly the same genes. So, if we're looking at the homologous pair of chromosomes, number 1, what you'll find on there is you will find the gene, let's say for eye colour from mum, and the gene for eye colour from dad. Remember, those genes could be the same. The alleles for those genes, the versions of those genes could be exactly the same, in which case you're homozygous for that characteristic, or the alleles that you have for those particular genes could be different, in which case you're heterozygous. So for example, you could have the eye colour on that particular chromosome or the eye colour gene on that particular chromosome from dad and it codes for brown eyes. You could have on the other chromosome of that homologous pair, again, the gene for eye colour, but this time the allele might be blue and that's what you've inherited from mum. So that's what a homologous pair of chromosomes is, okay? Those chromosomes are in homologous pairs because they contain the same genes. So what we're going to do next is we're going to take a look at those 4 different types of chromosome mutations. So remember, it's our translocation, inversion, deletion and duplication. And this little diagram here is just giving you an overview of what is happening. When I've got diagrams like this, the A, B, C, D and E, I'm referring to genes. Okay, so that is referencing to the genes that you might find along that chromosome. So we'll either see them as little banding patterns in different colours, or we might see them represented as letters. So the first chromosome mutation that we're going to take a look at is translocation. And this little animation is showing you what is happening in translocation. So you can see here in this particular animation what is happening is in our chromosomes, we've got two different chromosomes here. They are not homologous pair because they are different sizes. So clearly they've come from different parts. They're not exactly the same chromosome. In translocation, basically what's happening is a part of one chromosome is broken off. And then what is happening is that is moved and joined onto a different chromosome, not of a homologous pair, so a completely different chromosome. Now what you can see here is something called reciprocal translocation. So what's happened is that on the left hand side there, you can see that part of that very short chromosome that only contains those two genes A and B, the section of the chromosome with the gene B on it has broken off, and it has translocated and moved over to the second much larger chromosome. You can see it's now joined onto the bottom. The reason why it's reciprocal is because that larger chromosome, the same thing has happened. That section at the bottom of the large chromosome that contains gene number 7 has broken off and it has moved onto the shorter chromosome. So this is our translocation. So basically what's happened is a section of one chromosome has broken off and has become attached to another chromosome which is not its homologous partner. So think about what that means. Remember that in our homologous partners, the genes on the chromosomes are the same and they're in the same locations. So basically a whole section of a chromosome with certain genes on it is moving onto a completely different chromosome that didn't have those genes before, and those genes on that particular chromosome are moving over onto that different chromosome. So again, you can see that here in this picture here. So again, in translocation, you've got chromosome 1 and chromosome 2. Those letters are indicating the genes on the chromosomes. In this particular translocation diagram here, this is not reciprocal. So you can see our chromosome 1 that is all green is staying as it is. However, in our chromosome 2, the section of the chromosome that contains the genes S, T and U is broken off, and those or that particular section of the chromosome is now being joined onto chromosome 1. So it now has those translocated genes. Now the impact of this is that usually this will be happening when sex cells are forming. So when a body cell is undergoing a process called meiosis to produce either a sperm cell or an egg cell. Now, when translocation happens and we get whole sections of genes moved from one chromosome to another, it often means that the gametes are not viable. So that means that they, won't produce a functioning organism during fertilization. So it wouldn't be a viable sperm or egg cell. Now, the second chromosome mutation that we're going to take a look at is inversion. And again, you can see what's happening on that little animation. So here in inversion, basically what is happening is within the same chromosome, what is happening is a section of the chromosome is breaking off and then it is reversed and it fits back into that chromosome. Again, this is going to be an issue. So again, you can see that there in that little picture. So that section of the chromosome at the top where we have the genes D and F is breaking out, and then the D and F are being reversed and then stuck back into the chromosome. So we've got a whole section of genes here that have been reversed. So the chromosome is breaking in two places. The segment is rotating around before joining up again. Again, during the process in meiosis, when we're creating sex cells, if these chromosomes meet up with a homologous partner, which they would do, it would have to form a loop in order to pair up. And this has the impact in that if our chromosomes carry out what's called crossing over, they will have difficulty separating. And again, that means that those sex cells that are created in that process are not going to be viable. The next little chromosome mutation we're going to be talking about is deletion, and this is a bit more straightforward. So you can see here in a deletion mutation, basically a part of that chromosome is broken off and removed from the chromosome. So that means we're losing some of the genes. So there you go, you can again see that in that particular picture. So our chromosome is broken again in two places. The section of the chromosome containing those genes D and F is removed. Now, deletion has a drastic effect. All but the shortest deletions from a chromosome will be fatal because you've lost. You can lose quite a few genes, and obviously, if you're losing the genes, you're losing the proteins. You need those proteins to function. An example of a disorder that has resulted from a deletion of a chromosome is known as Cri-du-Chat, and Cri-du-Chat has occurred because of deletion of chromosome number 5. So in Cri-du-Chat, you can see there on the left-hand side, we've got a picture of chromosome number 5. People who suffer from Cri-du-Chat do survive, so this isn't a particularly fatal disorder, and you can see that actually it's the very, very short arm of the chromosome that is deleted, and there are effects of this particular mutation on the human involved, and what I will do is I'll link to a little video clip that explains what the effect of the Cri-du-Chat chromosome mutation has. Now, the last chromosome mutation that I want you to take a look at is duplication. So here, within our normal chromosome, what has happened is a section of the chromosome has been duplicated. So you can see here, that section that contains the D and E genes has been duplicated. So I've got that D and E twice. Okay, sometimes it might happen more than twice. You could be having it repeated 3 times. And here again, you've got some pictures of a duplicated chromosome. So this one on the right hand side is quite nice. This is showing you a picture of a chromosome. So you can see that black banding pattern has been duplicated. So again, just showing you our straightforward diagram. So you can see there our normal chromosome. What has happened is our section DEF has broken off and joined onto the homologous chromosome. So therefore, those genes D, E and F have been duplicated in one of the chromosomes of the homologous pair. So a segment of the chromosome's homologous partner becomes attached to one end or is inserted somewhere along its length, and therefore within that particular chromosome we have duplicated genes. Now, this can sometimes be positive because this may introduce new characteristics which may or may not have an advantage. Now, how duplication can be beneficial. So with our duplication mutation, what can sometimes happen is that because we have 2 copies of the same gene on the chromosome, what that means is that that second copy of the gene can be free from selection pressure. So what can happen is mutations can occur in the duplicated gene whilst the original gene can still be expressed and produce its protein. Now this means that that could produce, you know, for an organism, it's now has 2 proteins that are being produced, and that might increase the fitness and chance of survival for an organism. Now, a classic example is fish in Antarctic waters. They have evidence that they've had a gene that's undergone duplication. Now, that particular gene produced a protein that originally had 2 functions. So firstly, it was a digestive enzyme, but that digestive enzyme secondly had ice binding properties, and it stopped the growth of ice crystals. Now, often when we've got a protein produced, or a gene producing a protein like that, and it has dual functions, normally what might happen is through natural selections, one of those functions might be enhanced, but to the detriment of the other, so it might undermine the other. So normally, over time, that particular protein might primarily become a digestive enzyme, and therefore it wouldn't, you know, help with being an antifreeze protein in the fish's blood. However, when that chromosome gets duplicated, the gene gets duplicated, so you have 2 copies of the gene. And what that means is that then both genes could develop their functions, and this is exactly what's happened in the fish in Antarctic waters. Because that gene's been duplicated, one of the genes is producing the digestive enzyme, and it functions as a digestive enzyme. The other gene has actually evolved to produce a really well-functioning antifreeze protein. And when you look at them both, they have very, very similar gene sequences. So it's clear that, you know, one has evolved from the other through the, you know, process of natural selection. They've, you know, started to evolve their separate properties. So duplication can be important in terms of evolution. So that is our chromosome mutations. And remember, what I would like you to think about from this lesson is being able to identify those 4 different chromosome mutations and describing the impact that they have on the organism.